Welcome back to Lab at Home with the Museum of Life and Science. I'm Peregrine, and Catalyst volunteer Caroline is moderating the chat for us today. As always, we hope that you will help lead our program with your questions and thoughts and ideas in the moderated chat box. This is a really cool program, one of my favorites, and we've called it Primordial Soup. So I thought we might start there. When I say primordial, right, we are talking about way, 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 way far in the past. So for just a moment before we begin here, I want us to turn our brains back, our time machine way back, not hundreds or thousands of years, not millions of years, but billions of years ago. If we go back around maybe 3.5 billion years ago on Earth, that's when we're starting to see the very first living things. It's at least the first living things that left fossils for scientists to find. Some of the first living things were little types of bacteria called cyanobacteria. We'll talk more about them in this program. But cyanobacteria are some of the same bacteria that we'll talk about cultivating today in our experiment. So when we say primordial, we mean way far back in the past. And then when I say soup, I'm not talking about soup that comes in a can, called a soup because it refers to kind of this combination of different materials, probably in a body of water, all of these different things mixing together, and all of those different ingredients being what formed life way, way, way back, those billions of years ago. Thanks for putting that in the chat, Caroline. Cyanobacteria is going to be something we will talk a little bit more about today. So in our primordial soup, we are going to be focusing on something living inside of it. The things living inside of our primordial soup today, I guess it's not so primordial, but it is kind of soupy. The things that will be living in our experiment are the same things that we would be finding in primordial soup. That is to say, microorganisms and bacteria. So I want to ask all of you, my friends, when we're thinking about microorganisms and bacteria, tell me what you think about when I say those words. If I'm saying bacteria, microorganisms, what are they? What do they make you think of? What do you associate with them? What do you think? I bet you've definitely heard about bacteria before. This is great. My friend was saying miniature things, mini things in nature. Yeah, microorganisms is organisms, stuff that's alive, on the micro scale, the tiny scale. So things that are mini. How about bacteria? What do we know about bacteria? What do you think? I always think about bacteria as something that can get us sick. I don't know, that's something that I always think about. There's lots of kind of bacteria. There's tons and tons of bacteria all around us and on us and inside us. And it's true that some kinds of bacteria make us sick, but also some kinds of bacteria make it possible for us to live, especially when we're thinking about digesting our food. Bacteria is a big part of that process as well. So we're going to be taking a look at, figuratively taking a look at some microorganisms today, talking about some different bacteria and making a sort of home for them to live in. The home that we are making is called a Vinogradsky column. And I might ask Caroline to put that in the chat. A Vinogradsky column named for the scientist who developed it, Sergei Vinogradsky. He was a microbiologist who studied all of the teeny tiny things, these bacteria, microorganisms, and had this idea of not just isolating one and looking at it in a petri dish, but looking at a bunch of them all together to see how they interacted. So when we talk about our Vinogradsky columns that we're going to make, right, named after Sergei Vinogradsky in a column shape, which is to say kind of up and down, we're going to need a couple materials. Let me tell you about what we'll need. We're going to need some sort of clear container. That's going to be our column. I have a glass jar here. You can also use a plastic container as long as it's clear, because we want sunlight to be able to enter into this equation. We want sunlight to be able to get into our column. So the first thing you need is your clear column, and then you need a few other things. This is the fun part for me. I know when I go outside, My favorite places to be are around bodies of water, like creeks and ponds. I love places where I can stomp in the mud. How about you guys? I was really excited to go to the kind of little marshy area near my apartment for this experiment, because we are going to need to collect some water and some mud. So I have some water and mud here with me, right? My water is just water I scooped right out of this kind of still pond, kind of drainage body of water. And then I scooped some mud from beneath it. Now this is looking pretty soupy. Maybe not a soup I would enjoy eating, but it is definitely very soupy, very muddy. I know mine's got some sticks in it, some little pieces in it, and that's okay. We will work with those. I've got a few other things with me to make my column as well. I've got a source of two different materials. So all of us, right, we have to eat in order to live. We need water, we need food, and some of the things we need to get from our food are things like vitamins and minerals. The things that our body needs that we have to get from our food, right? And a lot of different types of microorganisms need the same things. They need different types of vitamins and minerals. The bacteria we will be talking about are ones that need a little bit of carbon and that need a little bit of sulfur and a few other things here and there. So I asked all of us to grab, if we're going to be doing the experiment along with us, I asked us to grab a source of carbon and a source of sulfur. You have a few different options, kind of some of the examples that we gave were good carbon sources are like dry vegetation, so dried leaves especially are going to be really good, dry grass. You could also use dry oats or cornmeal. That's another good source of carbon. I'm going to be using a little bit of paper towel because it has something else in here that I need. I'm going to use some paper towel for my carbon source, and I'm going to be using some eggshells for my carbon source. These are just eggshells here, and the reason that I used an eggshell is because my sulfur source is also from an egg. So there's a couple different ways you can get sulfur too. You can use Epsom salts, you could use plaster of Paris, or you could use an egg yolk. They can be raw or they can be hard-boiled. I figured I would have the whites of my hard-boiled eggs for lunch tomorrow, and I could use the yolk for my sulfur source today in this experiment. So these kind of go together. My carbon source here along with my paper towel and my sulfur source. The other cool thing about using an eggshell like this is that it has some other stuff that our bacteria might like to feed on, like calcium carbonate. You might like some of that stuff. This is a really good question I'm seeing from the chat here. We have a friend who is saying, why do we need carbon and sulfur foods, right? What do they do in there, in our little column? That is a really good question. Our different microorganisms, our different bacteria, and we're not going to list each one of them, but they need different things depending on the type of microorganism they are. We'll talk about it in just a second, what they need those to do. But we have one more material I wanted to mention, which is a lid for our jar, aka some plastic wrap. And then optional, but I always like to have it, is just a little rubber band for securing our plastic wrap lid. So this is a really good question. I might even ask if Caroline could put that in the chat for everyone to see, because this is such a good question about why we need these certain foods. What kind of things are going to be living in our column that need to eat these things? Let's go ahead and start up making their home, our Vinogradsky column, our primordial soup, and we'll talk a little bit more about it. This is really cool. I am going to go to my experiment cans. Here are all of the different materials we mentioned, starting with our jar. The first thing that we're going to do in our jar is we're going to take a little bit of this mud and we're going to scoop some inside. Now, before you scoop yours inside, if yours is anything like mine, It maybe has some lumps in it, or maybe got a little bit packed down. So make sure you're stirring it up so it's nice and soupy, right? Soupy primordial soup. Now, I don't know if you're like me and you gathered this pretty freshly and now you're stirring it up, you might be experiencing a smell. I'm stirring this up and it has a very strong smell. It's a smell that actually kind of reminds me of rotten eggs a little bit, right? This smell tells me that there are microorganisms in here that are doing a certain kind of work. There are microorganisms in here that we won't be able to see with our camera since they're so small, that are doing the hard work of breaking things down. So all of the stuff, right, that makes up this mud, little bits of dead plant matter, stuff like that, are being broken down by these microorganisms. And when the microorganisms do that, they release this probably less than pleasant smell. But for our purposes, it's good because we know that there's something alive in here and it is doing its work. It's eating its food and producing the byproduct of this kind of gross smell. So my mud is looking nice and soupy, and I'm going to scoop a little bit of it in here. We're kind of going to do some layers. I'm going to just scoop a little bit in here, a few spoonfuls. And if you're just kind of pouring your mud in, that's also okay. But I wanted to have a little bit of my mud on the bottom like this. And we're going to add a layer of paper towel. Instead of just sticking my paper towel in there, I'm actually going to rip it up into little pieces. That's going to make it easier for our microorganisms. So if you're following along, you can do like me and rip up your paper towel into little pieces. I'm just using a half sheet of paper towel. We don't need too much. We said that this paper towel is going to be a source of carbon for some of those microorganisms to eat. It's also a source of cellulose, which is the same stuff that you find in plants, which makes sense because paper towels, paper, comes from plants, comes from trees. This is going to be just some stuff for our microorganisms to chew on. It's going to be something that they need in order to eat. Carbon is something that we find in all living things, right? These are just particularly good sources of carbon and good sources of carbon that break down slowly, right? The thing about using a carbon source like cornstarch is that carbon would be eaten up, right? It'd be released pretty quickly. So I'm thinking if our experiment takes a little bit longer, we want to make sure that we have a source that is going to break down a little bit slower. I've got most of my paper towel in there. I'm going to scoop just a little bit more mud on top. Whoops. There we go. Just kind of 2 more scoops of mud on top of there. And I'm going to rip up the rest of my paper towel and put it on in there. This is perfect because when I spill mud on my on my mat here, I can just continue to rip it up and put it on back. So once I've got all of my paper towel in there, I'm going to add another ingredient. We said part of the purpose of the paper towel is to be a source of cellulose and of carbon, but we had another carbon source as well, right? Next up, we're going to add our eggs, our egg shells. But first, I'm going to add another layer of dirt here, another layer of my thick pond mud, and I'm going to stir it up a little bit. Is this looking like delicious soup to you guys yet? What do you think? So I'm stirring this up, just kind of making sure that all of the paper towels are soaked with mud. It now just looks like mud in there. And now I'm going to add my next source of carbon. So my eggshells here are already kind of broken up into pieces, but I'm just going to break them up into slightly smaller pieces and put them right in here. These are the eggs, these are the shells of a couple of eggs, but we really only need the shell of about 1 egg for a column of this size. The good thing about Vinogradsky columns is that none of your measurements have to be super specific. So if you have just a general idea of the different ingredients that you need, you don't have to measure out specific amounts, as long as we have a general idea. All right, so I'm sprinkling in some of my egg here. We said it's a source of carbon, and it also has another vitamin that's going to be really, another mineral that's going to be really helpful for some kinds of bacteria. And that's the calcium carbonate in here. So that calcium can be something that's really helpful for bacteria. All right. So I have crumbled up about an egg's worth, and I'm going to spoon on a little bit more of my mud, like this. And I'm going to do the same thing as before. I'm going to stir it up so it's well mixed in. This is some interesting looking mud now. It's got some eggshell, it's got some paper towel bits, it's of course got plenty of mud. And now we're going to add our one of our last ingredients here. I'm just going to grab the yolk of one egg, and I'm just going to kind of rip it in half and drop it in there. Now I had my yolk was a little bit softer. I didn't have a very crumbly yolk, so I'm going to use my spoon to stir it up in there. That's going to be very interesting. We said my yolk is a really good source of sulfur. That's actually, when we describe the mineral sulfur, we often describe it as something that smells like eggs. It's weird to think about a mineral, like a chunk of rock that comes from the ground smelling like an egg, but that is the smell that it has, and that is why eggs smell like that, is because of their sulfur content. Now I have stirred them up in here. I have got a very interesting combination of different foods. None of these sound like something I really want to eat, but then again, I'm not a microorganism. My friend had a really good question earlier about why do some of them need carbon? Why do they need sulfur? So there's lots of different types of microorganisms that are living in here, right? Tons and tons of these teeny, teeny, tiny things living in here. And there are different types that have different likes and dislikes. Some of them like a lot of sulfur, and they dislike oxygen. Quick question, my friends. What's oxygen? Can you tell me in the chat? Have you experienced any oxygen today? What is oxygen? Oh, this is great. My friend was saying oxygen is a gas that we breathe. Yes. Oh, exactly, my friend. It's air. So if you take a deep breath of air, our bodies need oxygen gas in that air. Now, some microorganisms like oxygen and they need it so that they can live, but some of them need a complete lack of oxygen. This column is going to help us grow kind of both kinds of these different microorganisms. So I'm going to add just a little bit more of my mud, and then I'll add my final ingredient. And we'll talk about the range of microorganisms that will live in here. So I'm just going to pour the rest of this in. All of our mud here. Nice fragrant mud with hopefully lots of microorganisms still in there. The smell at least would suggest that there are plenty in there. Continuing to stir that up. And now I'm going to take my water. I'm going to take my water from my natural source. This was just like a pool, a little retaining pool. And I'm going to pour it in here so that I've got a little bit of room for this water on the top. I'm not going to fill it totally full, but I'm going to fill it up to here. Now it becomes a little bit difficult for us to see where our soil line is, where our mud line is, but eventually this will start to settle down and we'll be able to see the layer of water above our mud. Now we were talking earlier about different things that microorganisms need, right? Not only this carbon, not only this sulfur, but we mentioned oxygen. I have a question for all of you. Do you think that there's a lot of oxygen down here at the bottom? What do you think? Do we think that there's a lot of oxygen going on down here? Yeah, I agree with you. I don't think that down here there's much oxygen going on at all, right? Certainly not any free oxygen, not any oxygen like we breathe in gas form, right? There's not a whole lot of oxygen under here because it's buried under all of this mud with all of this different stuff in it. And then the mud is even buried by some water, right? So if you are a type of microorganism that doesn't like oxygen, you would love to live at the bottom of this, here. Now, if you're a type of microorganism that does like oxygen, you're going to have a pretty good habitat to live in at the top. Hold on, I'm actually going to turn my face cam back here so I can point at it a little bit better. There we go. It's starting to settle a little bit. We were saying that if you are the type of microorganism that doesn't like oxygen, this is a great home for you down here. And up here at the top, even though it's waterlogged, there are types of microorganisms in here like cyanobacteria that will help produce oxygen when they photosynthesize. So just like plants produce oxygen, there are types of bacteria that produce oxygen. So they're helping to create, near the top here, they're helping to create this environment where oxygen-loving microbes can live. So what you get in a Vinogradsky column is this really interesting gradation of different types of microorganisms. The ones that like oxygen tend to be closer to the top. The ones that don't like oxygen tend to be at the bottom. The ones that need a lot of different, that need a lot of sulfur will tend to live at the bottom. And the ones that can't stand a lot of sulfur will tend to be at the top. Now, we can't see these microorganisms right now with our eyes, but the idea behind a Vinogradsky column is that if we give it enough time, these microorganisms will start to grow and grow in colonies big enough that we can see them. Caroline, do you think that you could show us a quick picture? Since we don't have, we can't quite do the time lapse with ours yet. Can I ask you to pull up some pictures so we can see some Vinogradsky columns kind of before and after they've had time to grow, right? So we see this is a picture from a lab where they were growing some Vinogradsky columns. It looks kind of like ours does, where it has just kind of some mud on the bottom and some muddy water on the top, right? But if we give it a little bit of time, it starts to grow, and the different types of microorganisms show up as different kinds of colors. The ones that we'll start to see on the tops, the ones that photosynthesize and produce oxygen like plants, they're going to tend to be really green. There tend to be kind of some reddish and purplish sulfur-loving bacteria that we see closer to the bottom. These are some really, really interesting growths that we see. I'm going to look at even one more that shows a little bit of a time lapse. So we can see that it takes maybe a few days for this stuff to start up. And then over time, over a number of weeks, we start to see these different layers emerge. I'm going to bring it back to my cam now. Beautiful. Caroline can go ahead and share some of the sources for those beautiful pictures. They're really cool resources for learning more about Vinogradsky columns. But you now have a great environment for a bunch of different types of microorganisms to live in. We do need to give them some time, and we need to give them one more thing, light. We'll want to store this somewhere where it gets not a lot of maybe direct sunlight, but plenty of light. So we're gonna go ahead and take our top, right, our plastic wrap, put that right on top, and I'm even gonna attach a rubber band there. Caroline was actually going to show us one that another Lab at Home viewer made. We did an episode on Vinogradsky columns back actually I think it was around last summer, and we had a viewer who did a really amazing job with her Vinogradsky column and sent us a picture. I would love, Caroline, if you could share that with us, what our Vinogradsky columns could look like in a matter of a few weeks. Let's take a look at that. Wow, that looks so beautiful. I loved seeing this. I could see all of the green photosynthesizing microorganisms there up top. Probably right, it's growing some algae, maybe some cyanobacteria. Then below that, you have kind of your reddish, purplish, sulfur-loving bacteria. It's cool because you can actually see some of her ingredients too. Like I think I see a little bit of the paper towel in hers. That's really great. All right, let's go ahead and bring it right back here. Thank you so much, Caroline, for sharing that one with us. That's so awesome. The last ingredient that our Vinogradsky columns need is time and a little bit of light. Now, these are sealed, but we know that a lot of the different microorganisms in here produce gas. So we'll want to keep an eye on it every couple of days to make sure that we're not seeing this start to kind of bulge as gas gets created in here. Another thing is, just like our pond mud, it may start to get stinky. I know that mine is probably going to get a little bit stinky, so I'm going to store mine outside. If you have the option to store it, maybe even right outside of your right outside of your doorstep, that's a great place for it to be. And this is an experiment that we can keep our eye on all summer long. So I would love I'm going to be really interested to see the progress of my Vinogradsky column. We love hearing from all of you. We have an email address that the Zoom comes from, the Zoom registration info comes from, that you're always welcome to send us pictures of. I hope that you start seeing some of these cool microorganisms in your primordial soup in the coming weeks. My friends, thank you for such a wonderful experiment today. Thank you for your awesome thoughts and ideas that you shared with us. All right, awesome. Thank you, my friends. I hope we'll see you next week for Little Lab at Home. I really look forward to that one with you guys, and we'll see you hopefully then. All right, see you later, my friends.